So here we're looking at the structure of DNA. DNA stands for deoxyribonucleic acid. Okay, and we know that DNA is present in all cells and contains the blueprint from which living organisms are made. Everything in the world is made of its own particular DNA molecules, so there's an infinite variety of DNA. When we look at the structure of DNA, we'll see why that's the case. So we know that a single strand of DNA is a polymer, and it is made up of just 4 different monomers. Today what we're going to look at is we're going to see what those 4 different monomers are and how they link together. Now, those monomers are called nucleotides. Well, so what's a nucleotide then? A nucleotide is actually made up of 3 different parts: a phosphate, a sugar, and a base. So here's my phosphate. Here is a sugar, and that's represented by a pentagon, and this is a base, like that. Now there's 4 different bases. One's called cytosine, one's called thymine, one is called adenine, and the 4th one is called guanine. Now you don't need to know the structures of those because they're given on the data sheet. Cytosine is often given the letter C, thymine a T, adenine an A, and guanine a G. On the data sheet they look like this: adenine, guanine, cytosine, and thymine. Now, this is a nucleotide, and 2 nucleotides can link together when an OH group of a phosphate on one nucleotide reacts with the OH group on a sugar molecule of another nucleotide to eliminate a molecule of water, and we're going to show how that happens. Then more nucleotide molecules can add on the same way to form a long polymer chain, which is a backbone of phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, with the bases coming out like this. And it's an arrangement of bases along the chain that can be in any order, and this leads to billions of different combinations of base order. That's why there's lots of different varieties of DNA molecules, almost an infinite amount. So what we're going to look at now is how the different nucleotides are linked together. So the first diagram I'm going to draw is a more simplified version of the nucleotides linking together. Now, to do this, what we need to do is we need to have a look at the sugar molecule here. Now, on a sugar molecule, which we'll have a look at in a second, there's actually an OH on there, like that. And on a phosphate group, there's also an OH. So what I'm going to do is I'm going to draw another phosphate group below this one, like that. And we know that contains an OH here. And that phosphate group will be attached to a sugar, just like it is above. Remember, sugars are represented as a pentagon. Bases are represented as a hexagon. And that particular sugar here would have an OH attached to it down here. And that OH will be attached to another phosphate group on another nucleotide, and so on. But I'm only going to draw 2 of these just to show how they're linking together. So we can see here that we've got an OH here and an H there. Now if I circle just those 3 atoms there, you'll know that there's 2 hydrogens and 1 oxygen. So that would eliminate a molecule of water, like this. So if we link those together, so we've got 2 nucleotides that are linked together like that. And again, this would link to another nucleotide below, and then again. And it's because we have 4 different base molecules that the structure of DNA becomes very unique to that particular organism. So, for example, if the base here was cytosine, so we just change that to a C, rather than just a general base symbol. Let's say that was adenine, A, that base, then that might be attached to another nucleotide. And again, that could be anything. It could be C, T, A, or G. Let's go for G, okay? And the process would just keep on repeating itself. And you have this long strand of DNA with a unique sequence of bases. Now, so every DNA molecule can be specified by the order of these bases. So for example, you could have C, C, A, G, T, T, C, A, G, G, T, T, and so on. A bit like a 4-letter code. And cells in the bodies of living things can read this alphabet. That's what cells do. They actually read this code. And once they've read that code, they can hold the instructions for making living things and replicating that code. That's how DNA replicates. Chemists can now sequence the bases in material from living things. So rather than cells, chemists can do this using techniques. They can read that code. And we call that DNA sequencing. We're going to have a look at the double helix now. So DNA exists as 2 strands held together by hydrogen bonding. So what we need to do is, we need to know how one base of one molecule can bond to another one. So in doing that, what we're going to do is we're going to have a look at our data sheet. So here's your data sheet. Now, if you haven't already done this, what I would suggest you do is print a copy of this out now for you to write on. In the back of your textbook is also a copy of this data sheet. I would recommend though that you actually print it out and write on it. So in the centre of the data sheet here, we've got our 4 bases. Now, we know that base C, cytosine, can only bond with base G. So G and C can bond together, and A and T can bond together. And we need to look at why this is. So I'm going to circle some atoms on adenine and circle some atoms on thymine. So I'm going to circle an H on adenine and an N on adenine. And I'm going to circle an O on thymine and an H on thymine. And what can happen is this H on thymine can hydrogen bond to that N on adenine. It's obviously not how you draw a hydrogen bond because I'm not orientating my molecules, but we're just annotating our data sheet here. and one of those H's on adenine will hydrogen bond to that O on thymine, like that. And we can do the same thing for guanine and cytosine. So I've circled 3 atoms there, and I'm also going to circle 3 atoms here, like that. So this hydrogen here can hydrogen bond with that O there. This H can hydrogen bond with that N, and that O can hydrogen bond with that H there. So what I'm going to do now is I'm going to draw these hydrogen bonds. I'm going to draw that molecule adenine hydrogen bonding with thymine, and that molecule guanine hydrogen bonding with that cytosine. And we can see straight away here that adenine can only have 2 hydrogen bonds, and thymine can only have 2 hydrogen bonds. Therefore, A will bond with T, and likewise guanine and cytosine can both have 3 hydrogen bonds, so G will bond to C. Clearly there's other lone pairs here that could form hydrogen bonds. However, they can't hydrogen bond to another cytosine molecule because of the orientation of the molecule. And that's what we're going to look at now. So when you're writing these out, please don't just copy what I'm writing down here. Have a look at the data sheet so you can see how I'm drawing these molecules. So I'm going to start off by doing that. And what I'm going to do is I'm going to draw a molecule of adenine here and a molecule of thymine there. And there's the beginnings of my hydrogen bonds. So that hydrogen there is going to come from adenine, which is going to be here. So I'm just going to put A there for adenine. And this hydrogen bond here is going to come from thymine, represented by this T here. You can see already the familiar hexagon shape of the base. And again, the familiar hexagon shape of the base again here. And the sugar, which we haven't gone through the structure yet. The pentagon shape here. And the phosphate group coming off the sugar. And likewise the same on this side. The sugar and the phosphate group, like that. Now you will be expected to be able to draw 2 hydrogen bonds between A and T. We're going to do the same thing with G and C as well. So this time we know that between G and C, guanine and cytosine, there's actually 3 hydrogen bonds. So I'm going to start off my picture like this. Okay. Sugar molecule. So here are 3 hydrogen bonds. Here, there's 2 hydrogen bonds here. So when we're drawing hydrogen bonds, what we need to do is we need to remember to draw the lone pair on the highly electronegative atoms. So we have a lone pair here, one there, here, here, and here. And we also need our partial charges as well. So we know that A has to bond with T and G has to bond with C. So it means that if two DNA molecules can bond together, forming a pair, but only if their bases are in a complementary order, an A on one strand matching a T on the other, and a G on one strand matching a C on the other. So if the strand of DNA had the base sequence of C, C, A, G, T, for example, then the other strand would have to have the sequence of G, G, T, C, A. So there's one strand there, and there's the other. The reason why that is because C can hydrogen bond with G, A can hydrogen bond with T, etc. But the shapes of the molecules are not linear like this. What happens is, in order to fit neatly over each other, the two strands here actually wind round each other in a shape of what we call a double helix. So if you imagine this is one strand, this will be the other. So the C on one strand would hydrogen bond with a G on the other by 3 hydrogen bonds. And let's say we had an A here. That would hydrogen bond with a T on the other via 2 hydrogen bonds here, like that. So the whole thing will be twisted together in a double helix. Okay, what we need to have a look at now is the more detailed version of how the nucleotides actually bond together. So when you consult the datasheet again, so on our datasheet, what we've got here is a phosphate group, glucose, and 2-deoxyribose. Glucose and 2-deoxyribose are examples of sugars, and this is your phosphate group. So what we're going to do now is we're going to look at how the phosphate group bonds to 2-deoxyribose. Remember the overall structure of DNA? of the nucleotide that makes up DNA. We have the phosphate group represented by a circle. So this is this. That is bonded to a sugar, represented by a pentagon there. For example, 2-deoxyribose. You can see the pentagon shape there. And that is bonded in turn to the base, represented by the pentagon there. So we need to know how the phosphate group bonds with the sugar and how the sugar bonds with the base. So the first thing we're going to look at is how the phosphate group bonds with the sugar. So I'm going to pick the phosphate group here in 2-deoxyribose as the sugar. So this oxygen here on the phosphate group will bond by a covalent bond. This is why I'm drawing a solid line rather than a dotted line, with that carbon there, that's CH2 on here. Now, in order for that to happen, we've got to eliminate an oxygen and 2 hydrogens. And we know what an oxygen and 2 hydrogens are. They're a molecule of water. So that is eliminated when they bond together. So I'm going to minus H2O like that. Now that's how a phosphate group bonds to the top of a sugar. Now we know from the structure of DNA that the next nucleotide, the phosphate group, will be bonded to this part of the sugar here, which is this carbon here. So what we need to do is we need to bond the top of the phosphate here to the bottom of the sugar. So in order for that to happen, this oxygen here bonds with that carbon there. It's this one in here. In order for that to happen, we must eliminate a molecule of water again. So for every phosphate sugar link you make, then you eliminate a molecule of water. So I've eliminated one molecule of water here via this one, and then another molecule of water here via that one there. Now we also need to know how the sugar is bonded to the base. So if we pick a base, let's say we pick cytosine here, then cytosine will bond through this nitrogen there. It can't bond through these three, can it? Because those three are involved in bonding to guanine on the other strand of the DNA. So it has to be via that nitrogen there. Now here, nitrogen's got 3 bonds. So what happens is, this hydrogen here is lost. That one there is lost when it bonds to the sugar. And it bonds this nitrogen here. I'm going to draw a line from that hydrogen. It bonds to that carbon there. For in order for that to happen, we need to lose that OH. So we've lost here an H and an OH. So that is a molecule of water again. Now all of these bases can bond in similar ways. By an H coming off an NH, for example here, and then that nitrogen there would bond to that carbon there. If it was this molecule here, it would be this nitrogen bonding to that carbon. If it was this molecule, it would be this nitrogen here bonding to that carbon. So that's how they bond together to form this kind of structure.